Hello, everyone. In this part of the course, we will make a mini workshop and we will illustrate how we can draw the cancer metastasis. Cancer metastasis is how cancer cells can go from one side of the body to another side, and this is responsible for cancer progression, cancer development, and this is one of the greatest factors that influence the mortality and morbidity of the patients. So in this part of the course, we will make a mini workshop. So we here can add new slides. So we have this first slide. We can like rename it. We can make it immunology cells or immunology cells. And in the next slide, we can add another slide and we can rename it as mini workshop. Okay, we can rename it as mini workshop. In this mini workshop, we want to draw how cancer metastasis can care. So we can go here and so cancer cells, they travel through blood vessels. They metastasize to other tissues through the blood vessels. So we have to find first blood vessels. Okay, the appropriate blood vessel that we can use to make cells look like they are squeezing and traveling to another tissue. So we found this one. We found this one. So let's reset zoom and we will find this part. Okay. So here we have the blood vessel, we have the endothelial cells and the outer layers of the blood vessel. Okay, so we have the endothelial cells and they are like lining the inner part of the blood vessel. This is a cross section. Sorry, this is a longitudinal section, which means we took a section through the current of the blood vessel, not through the cross section of the blood vessel. This means that this one is a cross section, which means we have like the blood vessel, this blood vessel, we had it as a tube, and then in the middle of the tube, we like got a cut section, we just look through it and we visualize its components, its linings. But here we show the lining in a longitudinal section. So we have the endothelial cells, and we can go and find like, for example, cancer cells. If you remember, we talked about the cancer cells that are squeezing. So we can like use this one. Okay, we can flip it horizontally, and we can make it travel like this one, or like this one, and so on. So they are traveling from the blood vessel. So let's make the travel here. We will illustrate like the cells are traveling from the blood vessel here, and they are going up, and then they travel through the distances between the endothelial cells. So we just would make some arrangements. This one looks like it is squeezing. Okay. Okay, we used it. We can use another one that shows that the cells were not yet squeezing. So we can start with this one here as it traveled from here, and Control V, Control C, Control V, or Control copy, and Control V, we copy and paste the icon in the same directions we're using. So these are identical items. So we put this one here, and then put this one here, and this travel. Then we like collect these cells outside here, which means they have traveled the distance and they like reach it the outside or the other tissue they were traveling to. Okay, so we have to use some arrows that show the process. So if you remember, we talked and we discussed this part. We said that we are going to use shapes and lines in another part of the course. So here we use shapes and lines. So we have here lines, arrows, inhibitors, dots, neurons, circulars, and so on. I'll use most of them in this part of the course so that we know how and when and where we can use this and every part of this one. Okay, so the lines are just for making borders. So for example, we can use this one as like we want to make a border for our design. So we can use it here. Yeah, as you can see. Also, we can split it and make an angle here. We use and grab the y dots. When we grab the y dots, we make an angle. When we grab the y dot, we make an angle. When we grab the y dot, we make an angle. What about the blue dot? The blue dot only edits the angle. Okay, so the white one creates the first angle. The dot or the blue one makes the hinge. Okay, so let me just go back in time. So as I told you, I've told you, we use the lining to just line and to make borders for our design. Okay. What about other types? Okay, we have different types of lines we can use. Also, we have arrows. So we will use arrows. We will use the curved arrow. Why not one of them? Because the blood vessel itself is curved. So we want an arrow that represents the curvature that we can find in the blood vessel. So we will use this one, for example, and we put it here. We find it like this. Then we will make a horizontal flip, and we will adjust its angle like this. Okay, we make it more labored. Okay, we grab this one here like this. We grab this one here. Okay, like this, and voila. Okay, then I don't think that the flipping of the arrow like this is good. I make it flipped horizontal, vertical, and then make it like this. I think that one is better because the cell will go from here to here and will travel to here. Okay, so we can use this arrow in this direction. Okay, okay. Then we will use another one, but a de novo one, and this de novo one we will make it like this. Okay, okay. And the cells have traveled. Okay, okay, that's it. Okay, so if you want make a review, if you want to make a preview, you can press here, and we have a preview, a short preview. If you want to make a presentation, you can press here, and you can see the whole presentation. Okay. Okay, I want to add some text to this part of the illustration. So we have talked about lines, arrows, inhibitors. Inhibitors means that the one that is on the left here inhibits what we are going to put after the mark here. So on the left side, we have the inhibitor. On the right side, we have the inhibited. Okay, the dots are useful in capturing the attention. What I mean by capturing the attention is that if we want to make an angle here like this, and then like this, we will rotate this one here. It shows the anatomy. What it means is that I can point to the cell uh, by using this one and making this line this, making it like this. And then, for example, I can add text here and write that this is cancer metastasis. Okay, cancer metastasis. I'll edit it, make this and this, and I'll make it somehow italic. Okay, and just making it like this. Okay, so I have used this dotted line to point to something in a wall, to point to something in the anatomy of something special. Okay, so also I can use it to point to the blood vessel, but I make it flipped vertically. And when I flip it vertically, I can point to the blood vessel like that. And I will make this a copy of this, and I'll write blood vessel. Okay, and just align this to the line. Okay, okay, okay. Also, I will take this one and like right here and make it align to the right and write metastasis. Metastasis. This is metastasis. Okay, this is what we call metastasis. Also, the blood vessel would contain blood cells, so we write red blood cell, and we like bring some uh, white blood cells. Okay, so yeah, we would put one here, put one there, and so on. We would bring another one in another direction to show homogeneity in the illustration. So like, but make sure to make them in the same size so that there would be no difference between them. So we would bring another one that is in another direction, or yeah, as you see, yeah, we can decrease their size. Okay, so yeah, so we remove this and take a copy of this to give us the same size. So we make it somehow correct. Okay. Also, the blood vessel would have some immune cells, as this is cancer, and surely the immune system has known there is cancer. So we will use cancer cells. So we will use immune cells. Immune cells are much more smaller. So we will like distribute them here, distribute this macrophage like here, distribute this A cell here, and so on. Okay, so we have composed an image of cancer metastasis. Okay, the most interesting part here is that we have used an icon that is composed of a lot of techniques to draw it. So we will have another slide to illustrate it better. I'll make it. I'll make a copy of it. So I'll take it copied. I'll paste it here. So how we can compose an icon like this? An icon like this is composed of a variety of copies of the same icon, and they are arranged. So we will see there are some of the cells that are on the back, some of them on the front, and some of them on the middle. Some of them are transparent, which means they are transparent, which means they can show the beneath layer or the underlying layer, their underlying layer. So we will go and take copy of immune cell of the cancer cell, sorry. So we will paste it here, okay? We will have different copies of it, and haphazardly, we will distribute them haphazardly, okay? So yeah, let me just finish it. Okay, okay, that's enough. I think that's enough. Okay, we will have some of them on the borders to make them transparent. So we will line the borders. As you see, I am just putting them on borders of the design. Okay, I am circling the design. So I'll then select them, press Control and select all of them. We will arrange them and make them send to back. Okay, so we have them send to back. And when we selected them, we have all of the edit options here. Then we will go to transparency and make it like 30, 40, or perhaps 50, 60, and so on until we reach the appropriate transparency we need. Okay, so we have them transparent. Then we will decrease their size. Okay, we will make sure that the icons on the front are somehow corresponding to the design and making it better. Okay, so we will rearrange them. Okay, so we will have another one here. Okay, and another one on the front here. Okay, then we will select all of them and decrease their size. Okay, but this one is not like this one. This one is much more perfect. How we can make it perfect? We should not make all of the transparency like this. We can make this one transparent by around 20%, okay? We can make this one transparent by around like 25, okay? We can make this one, of course, this one is 0 transparency, but this one would be like 20 transparency and so on. And we can increase the number of cells so that they look real, okay? So bring to front. And that's it. We can then Control G to arrange or to group them and just to make them like a bunch of cells or a cluster of cancer cells that have metastatized to another tissue. Okay, so we have talked about transparency, about grouping, and about alignment. And the next part of this workshop is that we need to export our figure. This figure that we have created, this one, we need to export it so that we can use it in our international journal. Okay, so we will go to shape first and we will make borders to the design. So we will make this like this, and then we will make its filling to be transparent. Okay, so we have only borders. We will go to the borders or the stroke and make it black and make it 2 and make sure it is not at the exact borders of the canvas. Okay, we will extend the canvas a little bit and we will make sure that it is extended beyond or beneath the canvas. Then we apply. Okay. Of course, we remove here the lines we have used before. Okay. So let me just remove this line and this line, and then we align it again. So we have borders of our design. As you see here, we have borders. It makes our design much more aesthetic. Okay. The next part is that we need to export our design. During exporting our design, we will encounter a lot of differences, a lot of different options we can use. Okay. During exporting our design, we will go here and then press export. When exporting, we need to export the first slide or the second slide or the third or the fourth one. We need a custom one. We need the second one only. So we will go to all slides and select custom and write the custom slide you want. I want number 2. Let's say you want 1 and 2. So I write 1, then 2. Okay, let's say you want from 1 to 3, so 1, then a slash, sorry, or a dash, then 3. So it will export 1 to 3. And when exporting multiple figures, multiple canvases, multiple images, it will zip them in a single file. So you have to extract the files after the software has make them a compressed file. Okay. So I want only the second figure. Okay. So as you see here, when I wrote the second figure, he or the software have presented a preview of the image. Okay. So I want a PNG or JPG or PDF. Our figure is very simple, so it's PNG recommended. If we have a very heavy photo, we choose JPG. If we want it as a PDF, we can download it as PDF. Okay, I'll stick to PNG for this time. Then I'll go to resolution and choose it to be 600 dpi, extra high resolution for some journals. Okay. Then if I want to publish it, I choose the publication permission to get the permission to publish it, and I'll stick here. If I'm not going to do that. Okay, you can preview your figure in a grayscale. So when we activated this, it is in the grayscale. Okay, I don't want it in grayscale. Then I press export. When pressing export, it will download and export our slide and ask us where we do want to download it. I choose downloads. Okay. Okay, and our image is now here. Let me show you the image. As you see here, our image is here. We have the metastasis, we have the blood vessel, the cancer cell, and we have to write all the details in our figure, of course. Okay, so as you see, it's very simple, it's very unique. After we have learned all of the tips and tricks we have been talking about for around 2 hours, like aligning, like how to arrange, how to design, how to make transparent, how to group, all of this we have like used in this simple figure. And it all starts with a simple idea. Okay. Okay. Let's just go back to our software. So we have like exported this one. Okay. So the next part of our workshop will be about the image extensions, the extensions of the images we can use in our design. Okay. So we have an interesting software, an interesting website we can use for this directly without the need of paying a bunch of money to other softwares. So we have an interesting website. Let me show you this website. So we share screen. So the first part, we go to Google and search for Conversio website. This is a website for file conversion. Okay, we choose the file we need to convert. Let's say I want to convert the file I have downloaded. So we go to downloads and we choose the file we want to download to convert. Our file was in the JPG. Okay, was in the PNG. Okay, we want to change it to like JPG. It is 1.6 megabytes. We start converting here. This website is free. You can go and make direct conversion without needing to without the need to even register or sign up for the website. Then we download it. When we download it, it asks us where we want to download, and we choose the location. Then we open the location, and it's also JPG. Okay, it's not, there is no difference, but I only show you how you can convert. Okay, okay. So convert more files. We go to like choose the same file. that we have worked on اللي هو which was the PNG, sorry, the JPG, and we choose another file extension like we choose the SVG, okay, and we convert. When we convert, we get an SVG file. This SVG file is, oh, some journals ask for an SVG file, okay? So you should be able to know where to convert your design. So as you see, the SVG file is a black and white, and we don't need this sort of conversion, but some journals ask for an SVG extension, okay? So we have talked about the PNG, the SVG, and the GPG. We have another extensions like Aviv and TIFF. Let me show you these types. So, okay, convert more files. And as we have talked, choose the file. Here you can find an extension that is called Aviv. We can search for it. It is Aviv. Some journals ask for this extension. And we have another extension that is called TIFF. This is the extension I was talking about, okay? That's just to be in the image and to know that there are different extensions we can use, different file extensions the journals can ask for. So you want all you are going to do, you will design your figure on your website, on the BioRender website, and you download it. Then you do the conversion required by the journals. Okay, so you don't have to suffer. You don't have to think more about the conversion process. You just design, you extract your file from the BioRender, and then you do the conversion you want for all your files through this website. Okay, so this is a very helpful website. It has been helping me for decades. This website is amazing. Okay, it is the best website on the internet for doing so. It is free, it is quick and user friendly. Okay, so the next part of our workshop today, as we have talked about exporting image and image extension, we will talk about the typography. Okay, so as you see here, I used this font, which is called Ropodo. Okay, usually I use 5 different fonts in all the softwares I use. In this course, in BioRender, in this website, BioRender, I use Roboto only. I don't use other fonts. I only use Roboto. But there are 5 different typographies or fonts I can use. Let me just write them here for you. So typographies I use. Okay. Number 1, I use Roboto. I use Times New Roman. Okay, Times New Roman. I use Arial. Arial is one of the best fonts. I use sans serif and serif. I use serif and sans serif. And I usually use also the Helvetica. So why I use only these 5 fonts? This is because most of the journals I have encountered during my research career ask for one of these fonts in your figure. Okay, so most of the journals, not topographies, it's typographies. So it sometimes make mistakes. So I use Roboto font. Here it is typography. Yeah, it is typography. So typographies I use Roboto, Times New Roman, Arial, Serif, Sans Serif, and Helvetica. They consider serif and sans serif as one font. Okay, serif or sans serif, they are one font. We also have Helvetica. So this is all about the typography section. Okay. The last part of our course, it is the first part of the advanced BioRender course. It is called the protein data bank. Okay. So let me have another slide. So as you see here, we have the icons, the templates that we can use, the brush. This brush, instead of having only, let me show you, instead of like we drag and drop here, this brush is all about increasing the efficiency of your work. So if you see here, okay, you can only use this brush if you want to draw a cell membrane. So instead of having only one icon of this, and then copying and pasting every icon of this beside each other, and then aligning them and so on, you can use a brush. A brush only allows you to just draw in the fastest way. So you draw, okay, let me just show you how you can use it. Okay, let's